Bismillahirrahmanirrahim. Hello, guys, and everyone. We are actually going to start off with the cranial nerve nuclei, right? Cranial nerve nuclei. To begin with, the development of the cranial nerve nuclei and the central and the peripheral nervous system, it actually begins with the neural tube, right? The formation of the neural tube. As you all know, like there was ectoderm, and then over here, if let's say we have the notochord, right, there would be inductive effect of notochord and the nearby mesoderm on this ectoderm, right? Now what would happen is that actually there would be formation of what? There would be formation of a neural plate, right? So there's actually a formation of a neural plate. Now what shall happen is that is actually there's actually, you see a groove formation. So a groove is formed right here, right? And as this region, if I can actually show you, as this region is getting pinched inside, as this region depresses, as you can see the elevation of this lateral margins, right? And with that, what we see is that there's actually, this one is notochord, and right here we have the formation of the neural tube, right? Neural tube, or this one is groove, and then eventually, I mean, this is not exactly a tube, but in the next stage a tube would be formed, right? Let's call it neural groove instead. That shall be more accurate. So then now let's form the tube. This one is the notochord and this is the neural tube, right? Eventually what will happen is that this one is the ectoderm and this is the neural, this one is the neural tube, this one is the notochord, right? So the neural tube is actually formed by ectoderm, right? If I can actually show you an enlarged section of this neural tube, because we want what is our topic? Our topic is actually the development of the cranial nerve nuclei. So if I can actually show you, let's say if this, for example, happens to be the neural tube, right? Right, for example, this one, if I can show you the different color, this was the neural tube, right? If I can label it neural tube, if this is the neural tube, here it is. All right, what is this? I've just shown you an enlarged section of this neural tube, right? Eventually what would happen is that let's say there's actually division of this neural tube into certain, that's not cool, let's make it a better one. Let's say this, I think it works. So this region right here is actually, so this region right here is actually what? This region right here is, let's say if this is present, let's say if this is actually present, if this is actually present dorsally, and this region, if let's say is present ventrally, we say that, what did we say? I told you that basically if this, let's say, is the we actually call these, we have different names for this. If this is the alar plate, and this guy right here is the basal plate, right? I used to remember this like A comes before B, so it has to be the sensory portion, and if B comes after A, this has to be the motor portion, motor component, right? How does this work? I mean, like if you're, let's say, just for the sake of understanding, if let's say this is the sensory, if there's actually a receptor, right? It would be a sensory nerve. Let's say this is the sensory nerve. It carries to the central nervous system, and then this comes and gives it the motor system. So this has to be the motor nerve, this has to be the sensory nerve, right? Okay. So alar plate, since A comes before B, so is the sensory portion, and B is the motor component. Another thing to remember is that the alar plate is also located dorsally. So this is dorsal location, it has a dorsal location, this guy has a ventral location, right? Just a bit of a recap. We learned that the cranial nerve and the structures of the central and peripheral nervous system would be actually be developed from the neural tube, right? From in this region, what we started is the development of the neural tube. Initially, there is an inductive effect of the notochord and the nearby mesoderm on the ectoderm, on the overlying ectoderm. There is actually thickening of a plate above the notochord. There is thickening of the plate above the notochord, and then there's actually formation of a neural groove, right? The lateral margins actually, if I can show you, is that the lateral margin would actually, since there is actually formation of this neural groove, the lateral margins would rise above, right? And eventually they'll move closer towards each other, and they'll actually pinch off, right? And as they, I mean, these guys are, and these regions actually meet. If these regions meet, so there's actually closure of this tube. And if there's actually closure of this tube, we, as we can see over here, we actually have development of this neural tube, and beneath that, as we had discussed already, this is the notochord which continues over here, right? Okay. By the way, if as I told you that there's actually closure of this tube, if this does not happen, then there's actually development of neural tube defects, right? There are problems with, I mean, there is congenital problems with, if there isn't any closure of this region. What exactly are those problems? Those are actually neural tube defects, and then, right, they can be, I mean, we would discuss them later on. And then, as we said, this actually, the neural tube actually forms into this alar plate and basal plate. But there's something also very important that I must mention, over here, is that this region that kind of separates the alar plate and the basal plate is called as sulcus limitans, right? Okay, as you can see that this area, I mean, okay, which is this one, this actually forms into the central canal of the spinal cord and the area of the ventricles in the central nervous system. But the problem is, as we learned about the fourth ventricle or the other ventricles, those ventricles aren't like evenly placed like this, right? Like if, for example, there's actually, let's say if we learned about the structure of the, let's say if this is the brain, so if this is the brain, and let's say there's actually cerebellum on the other side, and then so this actually has cerebellum. For example, if this is the fourth ventricle, so this must has the cerebellum at the base. But we learned in gross anatomy that the posterior boundary is formed by the cerebellum. So as we can see, one of these sides, so the alar plate is present over here. So that is exactly what happens. What exactly happens is that the alar plate actually moves laterally, right, to open space for this cerebellum over here, right? Let me show you guys in the next diagram. If let's say this is 1, 2, 3, and this 4. Brilliant. Let's say we should actually also, let's say if this is the fourth ventricle, right, and this one right here is the sulcus limitans, right? Okay, this one is the basal plate. Let's make the alar plate in the same color. Now what exactly has happened, right? We said that the alar plate should move laterally because we have to provide space for the development of the fourth ventricle. Let's say if actually cerebellum has to form here, cerebellum has to form here, so we have to move this extra regions laterally so that a ventricle can be formed, right? Now let me actually show you guys this. This is the latest movement of these alar plates, right? Now let's also label these guys. So these were actually alar plates. This was the basal plates, right? There's something important to discuss right now is that we said that, for example, this was the alar plate. It shall contain this. It is made up of the sensory portion. So this should actually contain the sensory nuclei, right? Let me show you guys if this is the alar plate. So it has actually 4 nuclei, and the basal plate actually has 3 nuclei. Okay, how about we actually, so these nuclei actually have some names, right? How about we actually, let's say, give them the names, whatever they are. For the basal portion, let's actually give them names. This guy, let's call this, okay, this guy right here is actually called as the general somatic afferent. Oh, apologies. So this is, sorry. Okay, this was the basal plate region. So this is the general somatic efferent. This one right here is called as the general or special visceral efferent. This one is the general visceral efferent. Okay. Now let's give them different color. As you can see, okay, this one, these region, these dotted lines, what are these? These are actually sulcus limitans. limitins, the ones that we talked about over here, the ones that actually separate the alar plate and the basal plate. Okay, what is the name of this nuclei? This nuclei is actually, okay, these nuclei, the ones of the alar plate and the basal plates, are actually kind of mirror images of each other, right? Of course, we have an extra one over here, but still the names are actually the mirror images. For example, if we had this one, this is the general visceral efferent, right? This is the general visceral efferent. So the guy over here is actually general visceral efferent. General visceral efferent. And what about this guy? This guy is actually special visceral efferent, and this guy should be general general somatic efferent, and this guy is special somatic efferent. Can you actually see the difference in their names? For example, these guys are called as, these guys actually have efferent in their names, right? But these guys have efferent in their names. How I used to remember this is that efferent and efferent. A comes before E, so which means these are the fibers that carry the information, right? And E comes after A, so that means these are the ones that carry these away. I mean, you also have this in when you were studying about, let's say if this is the Bowman's capsule, this was the efferent, sorry, the afferent, and this one is the efferent. Let's make a thicker one. So this was the afferent, and this one is the efferent. And if this is the Bowman's capsule, right, this is actually kind of the structure of the nephron. Okay guys, everyone, we would actually be studying these nuclei in detail in the next section. Till then, bye. Allah Hafiz.